Okay, good morning. So thanks for those kind words, Max. He was an old friend of mine, really sad news. So it's my privilege to introduce Rajiv Varshney, and also another friend of mine. I think we met in 2005 back in the crop mapping days. And if you've had a chance to read Rajiv's biography, I think there's two take-home messages that I get out of there is that he's very, very prolific in publishing and also very, very prolific in educating. So a very, very important researcher, particularly in my mind from the education perspective. Dozens and dozens of researchers have gone through his lab. So Rajiv started his career at IPK in Gottersleben for a few years, moved to work to CIMMYT in Mexico for several years, then spent the bulk of his career working in India at ICRISAT, and I think we're going to hear a lot of the research coming out of the ICRISAT days. But a year ago, Rajiv was lured away from India and has now moved to Murdoch University in Perth, Australia. And so Rajiv, I welcome you to the podium and give us some insights on some of the orphan crops and the African crops. Thanks very much.
Thank you very much, Daryl, for very nice introduction. And good morning to everyone. Let me put this thing first. Yes. Sorry. So, and good morning, everyone. I see large number of people. This shows that serious people are here and other people that are still sleeping or having the breakfast. Huh? So anyway, thank you very much for joining. It's really an honor and pleasure for me to be here and to share some of our work with this august audience. Before I start, I would like to thank all my well-wishers and mentors during last 20 years or so who have been an inspiration for me and have been guiding me and supporting me in different ways. Would like to mention especially name of my PhD supervisor, Professor PAG Gupta, and postdoc supervisor, Professor Andreas Graner. And of course, there are several people there sitting in this room, including Jeff Ellers from the Gates Foundation, Eric Dunca, Mark Sorrells, Ed Buckler, and I think there will be many other people. I may miss Henry Wade, so there are many other people. So I would like to take you way back in 2005 when I joined or when I attended for the first time the Plant Animal Genome Conference. Young boy, now lots of changes, got spec, got tummy as well, and there are a lot of differences. Earlier we used to have the fountain, now there is no more fountain. So only the senior people who have been coming here in the PAG, they know that about the fountain. But interestingly, many people that are still organizing their meeting around fountain, and there is no fountain. Huh? But anyway, so then at that time this was PAG13, and I really feel very excited to share some of the stories at the PAG30. And I took this journey with large number of colleagues from ICRISAT and our partners, and I feel really privileged and proud to say that we have worked for more than 180 organizations from 35 countries across 6 continents. My son, when he was young, he used to say, "Dad, you don't have any collaborator in Antarctica?" I said, "Not yet." So we will see that what happens. Anyway, now coming to the topic of this agriculture, and I think one key thing which I would like to share in this presentation, that how we have integrated the genomics technology in the crop improvement to contribute towards the food security. In this regard, I think agriculture and food, they are facing the global challenges at present. 3 important C's, for instance, COVID, conflicts, and of course the climate change. And when we talk about the climate change, then especially agriculture in the developing countries is adversely impacted, because in those countries agriculture is generally done in the rain or rain-fed agriculture. So, and we don't want to see this adverse impact continuously, we need to be prepared for that. On top of that, we all know that population is continuously growing. We already have passed, unfortunately, the mark of the 8 billion population, and I think that as the expectation is that in next 20 years or 30 years, we will be reaching this number 10 billion, so we need to be well prepared to feed additional 2 billion people or so. Not only that food, I think right now the most important thing is also the appropriate nutrition. So, and we know that at present also, 1 in 3 person globally is malnourished. And when we say malnourished, this can be different kind of undernutrition or overnutrition. So these are the issues, and this is not that while we are having these challenges new, and those we all know that I think around 50 or 60 years back, and especially countries like in Asia, there were many problems, and that this was the time that many countries, or because population growth rate was 3 to 4% in some of the countries, and country was at the edge of the famine. But at that time point, thanks to the leadership of Dr. Norman Borlaug, Dr. Swaminathan, Dr. Koch, and Dr. Yon Long Ping, that they were the hero, and they created the green revolution in the form of that transformational genetics and breeding by delivering the short height wheat varieties and rice varieties, and they really made a lot of change in that international level, but especially in the developing countries. And if you would like to see the impact of the green revolution during last 50 years, I think this is really very proud to say that now cereal yield has been increased for more than 240%. Cropping intensity has increased, and farming incomes have also been improved, a lot of modernization of agriculture, many good things. But one thing is that green revolution has also facilitated the liberal use of the chemical inputs in terms of the fertilizers, pesticides, etc., which is not good for the sustainable agriculture. And if you would like to see the consumption of the fertilizer around the world and just compare for about 50 or 60 years, you can see that we have reached this number from 52 million ton to 217 million ton. And this is continuous increase, though during last few years, we are being aware and we are working this direction. But now, if you would like to compare the world and with Africa, where we are always talking about the food security, Sub-Saharan Africa is having around quarter of that arable land of the world. But if you see that nitrogen fertilizer, etc., they are just using only 2.75%, because in that continent, we have not been able to deliver much fertilizer, which is not a good news. But at the same time, you see that cropping pattern, or for the major crops, yield advantages. The parts of the world like North America, Europe, or even in Asia, somewhere South America, wherever we have heavy use of the fertilizers, you gain the higher yield. Countries like in Africa, you had this issue, so there are always issues. But we are not saying that we need to push this fertilizer, but what we need to do, that we need to think that what may be the alternative ways to provide the enough nitrogen fertilizer, etc. And I think in this context, legume crops, they play a very important role because of number of reasons. They are the climate smart crops. You are having less water footprints. For instance, to produce 1 kg pulses, you need around 1250 liters water. At the same time, just to produce 1 kg beef, you need 13,000 liters, so means 10 times then. Same thing in terms of the carbon footprints. Legume crops, they're important for the nitrogen fixation, and there are very important crops from the perspective of the diversification when you have the cereal intensive cropping system. So legumes, they really play important role from the sustainability perspective, as you already mentioned. And I think the biological nitrogen fixation is about 20 to 22 million tons per year coming from these legumes. And on top of that, in many developing countries in Africa and Asia, they contribute a very important role towards food and nutrition security, especially from the protein perspective, because these legume crops, they got about 20 to 36% protein. And there are a number of legume crops. I will not discuss about every crop, but as Daryl mentioned, and I have been a big fan of Daryl from my postdoc time when he published this fantastic map of the consensus map of wheat. And based on these things, I developed at that time point a barley map. So thank you very much, Daryl, for introducing me here. Anyway, and then, yeah, so in this context, as he said, so I was a mapper at that time point, and when I moved to ICRISAT in 2005, we started to work on these crops, chickpea, pigeon pea, and groundnut, important crops in Sub-Saharan Africa and Asia. But if you see the crop productivity, and they are the rain-fed crops, this was about less than 1 ton per hectare. And this was the time, 2005, and so when I joined ICRISAT, this was a big challenge, that how we can contribute towards enhancing crop productivity. I was trained in IPK Garters level, learned the genomics approaches. So we were always interested that how we can contribute those genomics in this direction. But now, at the same time, and I think this was really, and I would like to share here, that 2005, when we had this rice genome sequencing happened through the International Rice Genome Sequencing Project. At that time point, together with Andreas, Graner, and Mars Sorrells, we provided a concept that how genomics can be integrated in the breeding program and how we can feed the world. And I think this was published in the 10th anniversary issue of the Trends in Plant Science. So this was one of the key things that we would like to put the genomics in the crop improvement. Now, if you think about the chickpea, pigeon pea, and peanut at that time point, we did not have the genetic maps. Forget the genetic maps. Even we did not have the molecular markers, crops like pigeon pea, 11 linkage group, but we had only 10 SSR markers at that time. So I think this was a big challenge, that how to develop the resources, genomic resources, and these crops used to be called orphan crops. So we took it as a challenge, and what we did, and I strongly believe in the partnership and collaboration, and then we started to work with different partners in India, in different countries in Asia, including China and Africa, and around the world in Europe and Americas. We developed several consortia, and then together with that, as a result of this partnership and advances in the technology, we were able to deliver the genome sequence of several crops, pigeon pea, chickpea, and in the case of peanut or groundnut, you are having this tetraploid species, so we delivered the genome sequence again together with the partners around the world for different species, pulse millet, and not only for ICRISAT mandate crops, but also we worked with other colleagues around the world, and we delivered the genomes for other dryland crops, which used to be called orphan crops in several times. So we were very happy that we got those genomic resources, and furthermore, when we do the genome assemblies or so, we can always predict the genes, but we do not know their function fully. So what we did, that we also developed the gene expression atlases for each of these 3 legume crop in chickpea, pigeon pea, and peanut, and we use the gene expression atlas to understand the mechanism of the traits. For instance, in the case of chickpea, we use the genomic resources, these approaches and other approaches I will discuss further to understand the basis of drought tolerance. In the case of pigeon pea is one of these unique legume crop where we have the cytoplasmic male sterility based hybrid breeding system. So then how we can understand this mechanism of the cytoplasmic male sterility. In the case of peanut, we understood that mechanism of the oils, fatty acid biosynthesis pathways, etc. So these were really important resources. One more thing, that when we developed these genome assemblies, at that time point we were using the short sequencing reads. We started when Illumina was just 36 base pair reads, or even the Solexa sequencing technology, GA1, etc. long back. So those genome assemblies, they were not very high quality, but during last few years, we also use the modern sequencing technology, including Hi-C, and nowadays we are using PAG30, Hi-Fi, so as many other people are doing, and we develop those chromosome length genome assemblies for all these legume species. And these high quality genomes, they helped us to understand the evolutionary events that shaped the modern days legume crops. At the same time, different legumes, they have the different or unique traits, and what we did, that these data also provided us, or they helped us to understand the basis for the unique traits. For instance, in the case of peanut or groundnut, the mechanism of the nitrogen fixation is different than other pulse crops. So I think this was really very helpful. And furthermore, and this is an example from pulse millet, for instance, in collaboration with Corteva, that earlier we had the stiff genome, but now you can see the platinum standard reference genome for the different pulse millet varieties. So a lot of things are happening. Now, these genome assemblies, they were not just helpful for us to improve these crops, but they also helped to improve other crops. For instance, in the case of pulse millet, which is very drought and heat tolerant species, and pulse millet can provide the seeds even at the 42 degrees Celsius temperature, and where the major cereal crops like maize, rice, or wheat, even temperature goes more than 36 degrees Celsius at the flowering time, we don't have those, we don't have the seed setting and other thing. And the analysis of the pulse millet genome together with other cereals provided us about some 384 expanded gene families, which are coming from these pathways like terpenoid backbone biosynthesis, cutin, suberin, and wax biosynthesis pathways, that why the pulse millet is much more drought tolerant and heat tolerant, and can we use those resources now to improve the other cereal crop. Same thing in the case of pigeon pea, which used to be called orphan crop, and soybean is an industrial crop. But now you see that in soybean we have this issue of that Asian soybean rust, and there was no natural variation. And way back when we were developing these genome assemblies, and Jonathan Jones from TSL, he and we discussed something, and then they used that pigeon pea genome to clone the gene or to identify the gene, and this was the pigeon pea gene, which confers full resistance to Asian soybean rust. And at later stage, they were able to deliver the varieties through some companies, and now some of these varieties, they are already reaching to the farmers in South America. So this is the beauty, that many times people say that this is minor crop, you don't need to develop resources here, who will use, who will cite the paper. But here, these are the example that even we didn't know at that time point, but crops like pigeon pea can be helpful for even the soybean. So anyway, and once you got these genomes, next question is, as many of us have been asking for last few years, whether sequencing one genome is good enough, and answer is no. Why? Because if you got the genome assembly for cultivar 1, but you are having some unique genes in cultivar 3, and if you don't have the genome assembly for cultivar 3, and you got just on cultivar 1, then you are missing a lot of stuff. So I think there is a need to sequence more genomes, and so that we can understand, we can have the genes across the species. We extended this concept further at the genus level, and we say, for instance, in the case of chickpea, if you got 8 different species, can we develop the pan genome for each species, and then we develop a pan genome across the species, and we call it super pan genome at the genus level. And in this direction, so we have started to working on this direction, and we believe that when you are targeting that land races or crop wild relatives, and you are developing the pan genome, then you can provide the genes or markers or haplotypes associated with the climate resilience traits, because in that land races, you got those genes during the course of the domestication or breeding, you have lost several of those genes, so you can do that one. In the crop wild relative, again, sometimes they are not crossable with the cultivated species, but now with the power of the technology like gene editing, etc., it is possible to go for the de novo domestication or to improve some of these traits back to the cultivated species. So with this promise, we started to sequence large scale germplasm, including in the case of pigeon pea, for instance, we targeted about 292 pigeon pea lines coming from 23 countries, 429 chickpea lines coming from around 40 countries. And another good thing what ICRISAT and our partners had, that they had really good capabilities, capacity for undertaking large scale phenotyping. And this was really very plus point, because nowadays you can generate sequence data very fast, but phenotyping is very important. So what we did, that by combining these data together, we were able to identify the genes associated with the traits of interest for the breeders, and especially for the climate resilience traits, and like for instance yield under drought and heat stress. We expanded this study further, and in the case of chickpea, where we have the total accessions, about 20,000 accessions. When I was at ICRISAT, my dream was to sequence all 20,000 at the whole genome coverage, and I'm happy to mention we already completed about 10,000 when I was leaving. But in this paper, we reported the sequence of genome variation map based on the 3,366 genomes, and in addition to understanding the genetic diversity, we developed the pan genome, which provided about 1,600 additional genes. Lot of stuff, I will not go in the detail, but one thing is that we also understood the center of origin of chickpea based on this data, which was in Fertile Crescent, and also the migration routes, because now chickpea is grown in Africa and Asia, and there were 2 pathways, migration routes. One was to South Asia via the Central Asia, the other one from that Fertile Crescent to the East Africa, and at later stage this went to that New Worlds or so. From the breeding perspective, based on the phenotyping, because we had these 3,000 chickpea data, and we also phenotyped these 3,000 chickpeas at 6 different locations for 2 different years, and for large number of agronomy related traits. And when we put this data together, not only the jivas, but we went one step further. We identified the haplotypes and superior haplotypes. I will talk about this thing later. And I think this is a good resource now for enhancing crop productivity. So this was some of these output from this study. Our interest was always that how we can use the genomic resources for the crop improvement. It's good to develop those resources, publish the paper, not for enhancing your career, but of course that for next generation of scientists this is good. We wanted to publish this work so that this becomes available to the entire world. But key thing was not publication, rather that how we can put these things in the crop improvement. And when you talk to the breeders, they're always interested that what is the cost of the genotyping, whether we can afford these things or not. And with this objective, we developed a range of the genotyping platform, because one size does not fit all. Sometimes you are having the breeding population, sometimes germ plant collection. So we developed a range of the marker genotyping platform, and we are not saying that you need to use every platform for every perspective. Based on the cost benefit ratio, you can use this platform. So based on these things, we genotyped different mapping population, biparental mapping population, multi-parents mapping population, including MAGIC, nested association mapping population, germplasm collection, etc. And phenotyping was always an issue, but then together with our colleagues at ICRISAT and partners around the world, we also address this issue, and we have phenotyped this collection for number of traits. To make long story short, during this time, we put all these things data together, and as Daryl mentioned to me, like as a mapper, so we mapped lots of traits, and I think about 20 to 50 traits in each of these crops, which is a big thing. This is not coming from Rajiv Varshney alone, but with large number of partners, which has provided our leadership and support to the different colleagues. So what happened that in the case of chickpea now, you got that markers of maps for drought, heat, salinity, ascochyta blight, the different biotic and abiotic stress constraint. Same thing in the case of groundnut and in the case of pigeon pea. So we mapped those traits. We had some information that where are the markers, which chromosome, what kind of flanking regions, so we got these things. So next thing is that we asked the breeders, and not only at ICRISAT, but different organizations, that let's translate this genomics research. We would like to see, we would like to enhance the crop productivity. And in this direction, as I said earlier, so we optimize some approaches. At that time point, marker-assisted backcrossing, marker-assisted recurrent selection, genomic selection was just coming up at that stage. So we started to work that well for which trait, in which species, what we can do. And when we started to talk to the breeders, they always say, this is very fancy, very good. But now, what happens that what will be the cost? And as I said earlier, we asked that, what is that when they say? For my line, when I do the phenotyping, field evaluation, for any line in 3 replicates, 3 locations, etc., I don't spend more than $15 per line. Can you bring your cost down? And at that time point, as you talk about the DNA extraction, $2 to $3 per sample, markers very expensive. So this was one issue, the genotyping. Second was the capability and capacity, and when you are talking about the breeding programs in Africa and Asia, you should not expect that everybody is, or they are having the labs of that standard or the capacity. So these were the 2 challenges, and what we did that we tried to address them. And here one of them is for one project, high throughput genotyping project, which I led from and to work together with 8 CG centers with the strong support from Bill and Melinda Gates Foundation and Gary Atlin, who is not here in this meeting this year, but we used to come to every year and we used to discuss. So I think this was that very good project. And what we did together with that service provider like Intertech and with that providing our resources, this project delivered more than 1,000 SNPs for more than 100 traits for about 18 crops. And that cost, we brought the cost down at the level that you can genotype any line for 1 to 10 SNPs in $1 to $2 per sample. And then when we went to the breeders, that now you got the resources, so cost is not an issue, right? Next thing was again this capacity or so, and I feel so happy and proud, and again thanks Daryl that you have recognized that you mentioned. Another thing what we have been doing at ICRISAT, the training the breeders coming from Asia and Africa, not that how to generate the data, but rather how to use these data in the crop improvement program. And we feel so happy that we trained during last 10-15 years around 485 scientists and 365 from India, but you can see that around 150 from the Eastern Southern Africa, 40 from Western Central Africa, and about 31 from South America. And I think this was a big boost to the breeding program. And then we did work like a hand-holding kind of thing, work together, not that we are instructing, guiding, or something's working together. And then breeders started to work together, and here I would like to highlight some of these success stories. And here, okay, so another thing is that not only training, and those of you who have been working with us, you know, when I was at ICRISAT, we were hosting different kind of conferences. Objective was to bring the experts from outside and then bring young generation, the scientists from Asia and Africa in those meetings and keep them updating with the new technology that where to move and how to move. And this really helped us a lot to promote those genomic technology, genomic breeding in the developing countries. So anyway, so based on these things, breeders started to work, and here I would like to highlight some of these success stories that how we were able to mobilize the genomic resources to develop the improved varieties even in so-called orphan crops in the developing countries. For instance, here we have this drought-tolerant chickpeas. My colleague, my good friend, Dr. Bhardwaj, who is here sitting in this meeting room as well, very forward-looking breeder and from Indian Agriculture Research Institute. So he and his team from IRI and also some other colleagues from the Indian Institute of Pulse Research of ICR worked together, and in very short period of time, we developed several lines. And in India and many other countries, you are having very rigorous variety release pipeline because you need to have these improved lines, need to go across the country, You need to compare the yield with the topmost variety at that time point, select then go next cycle, very rigorous breeding pipeline or so. But we are very happy to mention that several improved drought tolerant. I'm mentioning about drought tolerance. Earlier stories for the molecular breeding, you will see many times in the biotic stresses where that you are having those traits controlled by 1 or 2 or few genes. When you are talking about the complex trait like drought and having the varieties having the higher yield, more than 18 to 20%, this is really a big thing. Because, and we were successful because we identified a QTL hotspot. I always call it that QTL hotspot in chickpea because this contains the QTLs for more than 20 traits. But anyway, so then through this program we had PUSA JG16, IPCL 414, PUSA chickpea 10216, PUSA chickpea 4005, they were coming from the drought tolerant. Same thing for the Fujiram wilt resistance. This is another trait. Here, Dr. Mannoor is another chickpea breeder, and he developed a variety called Annegiri way back in 1970s, even before I was born. And breeders, as you know, that they love their varieties more than they love their children. And yes, so, and I think just before his retirement, and he came to one of those our training course, and he says, Rajiv, you are talking, everything can be done, genomics looks like a magic. And now my Annegiri variety is not being promoted. Farmers are still growing, they love that one, but the yield is very low because this is susceptible. Can you help me? Can you make my Annegiri variety as a Fujiram wilt resistant? We say, let's see. We worked together, and then we developed this new line. This was released with the name as Super Annegiri 1, and this became again popular in the farming community. It started to deliver the yield about 1.9 ton per hectare. And Dr. Mannoor, when he retired and he became very emotional. He says, Rajiv, I never saw the DNA by my eyes. I say, you cannot see the DNA by your eyes. But anyway, he wanted to say, and then I'm very happy to see the DNA product in my field. So this is something that like you can encourage, you can inspire not only the young scientists, but even the breeders who have been working for a long time, that they feel very happy that how the genomics technology, they are helping their crop or their varieties. Similar is the case for the Ascochyta blight resistant, where we had the breeder Dr. Sabjit Singh, Dr. Inder Singh, and Dr. Shyala Bindra, and they are coming up with this new variety of the Ascochyta blight resistance, because in the Punjab state we still have the Ascochyta blight. Now, not only in India, we had this several projects funded from the Gates Foundation, one of the tropical legumes project, I will mention about that thing later. But here, these are the cases like Dr. Asnake Fikre, chickpea breeder, another breeder, and this is Guise Nirma. And then these breeders, we work together, they release these two varieties, Gale2 and Girar, and which is also having around 3.8 tons or 2.1 ton per hectare. And again, much more higher yield advantage, so two varieties in Ethiopia. Same thing in Kenya, together with our colleague and collaborator, Dr. Paul Kimirto and his team. And these students, at that time, they were students, they were coming to our lab, doing the genotyping work at ICRISAT, taking those lines and making those crosses in Kenya. And now some of these lines, they're in the advanced stage of the release in Kenya for drought tolerance. In the case of Tanzania, we had several lines from the marker-assisted back crossing, and here we used the different approaches. We took the farmers and we used something like farmers' varietal selection, and in those cases, farmers, they really loved those MABC lines and some of these lines, and this is Joseph Joachim and Asnake Fikre. So these people now they are taking, or some of these lines, they are going again as in the form of the release in Tanzania. Next point is that you can release the variety, but now how they are making the impact or how they are going to make the impact. So we need to see this pipeline as well. In this case, I think there are a lot of role from that organization, seed extension agency, etc. In the case of chickpea, in India, I would like to mention that like Dr. Bhardwaj and his team, they are working with the different seed system agencies or so. They are developing the breeder seed or so in such a manner that in next 2-3 years, they should get enough seed. And whatever they can do, and their plan is that they are already producing the seeds in such a manner that these two chickpea varieties, they can cover in next 3 years around 30,000 hectares and 43,000 hectares. We did some analysis further, even with the assumption of 70% adoption, around 60 or 70% of the realized yield and the potential, then what is the additional benefit farmers will have? And when we did the analysis, even with the very conservative estimate, we believe that about $20 million will be added to the farmers from one variety and the another set of the $61 million. And we have done the similar kind of thing in the case of peanut. In peanut, you are having the different issues, but I would like to just mention about this fatty acid biosynthesis pathways where you are having In general peanut varieties, they got about 30% linoleic acid, not good for health. Oleic acid is good, so what we did, that we developed these oleic acid lines, and India released the first set of the high oleic varieties, and again, like Graner 4 and Graner 5, we are moving further that how we can cover more areas. The prediction is that with these two varieties alone in next 3 to 4 years, you can add this kind of more than $300 million and $89 million to the farmers. In the case of peanut again, for foliar diseases, Agriculture Minister of Karnataka state at that time point, and they also released some of these varieties in Karnataka state for the foliar diseases, and several other foliar disease varieties, they are coming from that India. So these things are happening, and I can keep on going and on, but this is not the idea. Idea is that basically to show that how we were able to deliver these varieties. Now in the Ghana, and Richard, he's really fantastic breeder, and in his field now you can see this foliar disease resistant variety, also the high oleic varieties, and they are also in the pipeline in the case of Ghana from Savannah Agriculture Research Institute. In the case of pigeon pea, so we got these Bhima varieties. So you can see that during last 10-15 years, we started from scratch, developed the genetic maps, identified the QTLs, introgressed them, and even in those cases or so, we already are having more than 20 varieties coming through integration of the genomics in the breeding program. Now the next thing is that even if you are having these varieties, irrespective from which method they are being delivered, I think this is very important, the developing countries' perspective, that they should be reaching to the farmers in the real time. Farmers should be using good agronomy packages to realize the full potential of genetics. Sometimes you are having these issues of the post-harvest losses, and if you would like to have more money to farmers, they need to be connected to the market, and if you can have the value addition, then I think that this will be very beneficial to the consumers as well as to farmers. To address some of these issues, and I would like to highlight very quickly a few examples from another big project, Tropical Legumes Project, and this project was run for about 13 years, $67 million initiative from Bill and Melinda Gates Foundation. I had a privilege to lead this project for about 7 to 8 years as a project principal investigator. I had very capable project coordinator like Chris Ojibo and Emmanuel Monio. Before that, we had Sadek Abate. Before me, Dr. C.L. Goda was the PI. And we had very strong support from Dr. Jeff Ellers as a program manager for this project. And this project was long back before Jeff. This was initiated by David Worthington. So, and this project was coordinated by us at ICRISAT, but implemented by 3 CGR centers, including CIAT and IITA. We have worked in 13 different countries, Kenya, Ethiopia, Uganda, Tanzania, Malawi, Mozambique, and Zimbabwe in the Eastern Southern Africa, and Mali, Senegal, Burkina Faso, Ghana, Niger, Nigeria, and Western Central Africa, India, and Bangladesh. We worked on 6 different legume crops, and what we did that this project, because this was big project, many partners facilitated development of about 300 varieties. We worked on all those traits which I was talking, and also we also focused on the market preferences like fodder at home quality, fast cooking, palatability, high oleic, etc. The another thing which through this project, what we did, that we wanted to help the breeding program so that they can deliver the higher genetic gains, and we worked in this direction, and we also, as I said earlier, so not only in those few crops, but across the crops, we did this kind of thing. Third thing is, and yeah, so these are some screenshots, you already have seen some of them, but one other key thing from the seed delivery platform system. In countries like in Africa, this is the big thing, and when you are talking about the cell-pollinated crops. So this was key thing. So what we did that we developed the sustainable seed delivery platform. We engaged more than around 2,000 seed producers coming from the public seed companies, private seed companies. We addressed the issues of the adoption barriers. We created the seed demand and raised variety awareness. We also ensured that seed system is the gender sensitive. This is also a very important thing. In summary, this project delivered about 300 improved varieties, 397 ton certified seeds, 4.4 million hectare area was planted, and about 4.9 million ton grains were produced with the worth of $2.6 billion. And based on the economic analysis note from Rajiv Varsne, I think this project touched the lives of about 22 million farmers. Again, capacity building was key thing in Africa, and we trained more than 52 PhD scientists, and then they are now leading those kind of program in the different countries. In terms of the impact, I don't have time, but all these data in public domain, but what I would like to mention, that in the different countries for the different crops, you had the crop productivity enhanced, you had the area enhanced, you had the seed production higher. So all these things. And just yesterday when I was talking to Jeff, and I will come back to it in a minute, and we have documented all these achievements. So this is not that everything is rosy picture or so, at the success story, we had lots of challenges. We documented those achievements and challenges in the form of the books or so. But one thing which I was saying that yes, Gates Foundation, they also did this analysis through the independent agency, and this is Mathematica. They developed this report, they submitted this report to Vance School Policy Analysis and Research. I have taken the snapshot, and they have done this ROI analysis of the TL investment. And if you see the financial rate of return, they say this is the 16. So benefit to cost ratio across segment is 16. This means that every dollar invested in the tropical legume project generated nearly $16 in value for smallholder farmers. This suggests that the full portfolio of TL investment had very high social returns relative to the BMGF's level of funding. We feel very excited, very impressed with these kind of reports, and I think that this project impact, they were also recognized by Africa Food Prize to ICRISAT. I see Daryl is here, but very quickly I would like to mention few quick points. Now, what is the future? I think that when we are talking about the genome sequencing approaches, we are already reaching the haplotypes thing. I think this is the time now that we can use the haplotype-based approaches, genomic prediction. Other thing is that how we can select the parents in such a manner that you can have the optimal contribution selection from the parents, and the gene editing, and I think by using these methods, we can deliver the varieties in much more faster manner. I already mentioned, but good thing is that now for majority of the crop species, we got the reference genome. We need to see that how the sequencing and genotyping, this can be democratized, and we are moving in the area of pangenomics, haplotypes, etc. Here, and I'm being big fan nowadays for this approach. So now when we are got a lot of sequencing data for each gene, you can identify different haplotypes. Next thing is doing the analysis with the phenotype, and then based on these things can we identify the superior haplotype for the different genes. We are going further that how we can use the artificial intelligence approaches to identify different kind of haplotypes, and even not this one, that when you combine these things, what will be the impact on the yield, etc. And based on these things, you can analyze that what kind of haplotypes you are having in the varieties. If you are missing some good haplotypes, you can convert them in the superior haplotypes. We did some work in the case of chickpea, and we had some 56 promising lines which had a superior haplotypes. We are very happy to mention that we had even the visit of Honorable Prime Minister Modi in the chickpea field, you just imagine. So I think that this is the way that these superior haplotype breeding, etc. can work. And furthermore, for the genomic breeding, I mentioned about the optimal contribution selection, different approaches of the genomic prediction, and when I was at ICRISAT, we worked something in this direction in the different legume approaches, and I think this is going to be helpful. Last point which I would like to mention, that we are talking nowadays more about the gene editing, but for that, we really need to have the gene with the causal effect. And this is one of the key things, and now I think many organizations, many of us working on this aspect. And what we are doing, that we are using the systems biology approach, where we are integrating these approaches together, and we delivered some of these results in the case of pigeon pea, where we combined these different approaches, and even for the fertility restoration, which is a key thing for cytoplasmic male sterility, we found some genes in the auxin biosynthesis pathways which are responsible for making some pollen grains viable, some of this sterile. So these kind of approaches are required. We are moving towards now spatial transcriptomics, and I always compare this spatial transcriptomics like fruit chart. So if you are having the smoothie, this is like that you are enjoying the fruits, but you don't know what is there. And here you know everything that, well, where, which gene, what kind of expression, what kind of correlation. So I think these approaches are going to be helpful. In my view, that this marker-assisted breeding, they are moving now in this direction, haplotype-based breeding, genomic prediction, and also that gene editing. So this is some of the thing. We are using some of these approaches now at the new place at my center in that Murdoch University, Center for Crop and Food Innovation, where more than 15 research group leaders, they are affiliated to this center. Our center is having the different themes: crop production and quality, nutrition and food, crop protection and microbiome, crop adaptation, digital agriculture, path to market, and also science management, etc. And each theme is being led by very capable scientists and research groups also. And here, my own research group is working now on wheat, legumes, and nowadays on the horticulture crop. And if you would like to see this platter, we are providing the carbohydrate, proteins, and also the micronutrients and vitamins, etc. in the form of these things. In terms of the approaches, now at Murdoch, we are using more or less the same approaches, so like pangenomics, computational genomics tools, trait discovery, and all these things. And let's see that PAG, they took 17 years for me to give this chance for the plenary. Maybe that they will not be taking another 17 years, and then maybe next time I will be discussing some of this research output from the Murdoch. Anyway, so to summarize, I think the crop diversification is essential for sustainable agriculture. And in my opinion now that this is the time that many of us already started to use the artificial intelligence, machine learning and simulation approaches, that how they can accelerate trait mapping, etc. In my opinion that now the GAV1 and GAV 2.0 approaches, they are very important for developing new improved varieties. Finally, from the developing countries perspective, I think the financial support and capacity building is really important when you are talking sustainable agriculture. And finally, thanks to all the colleagues at ICRISAT and some of my colleagues like Anu, Manish Pandey, Manish Rodkiwal, and some of these, Amir, Sandeep, they are already here in this conference, but there are large number of people, very capable people, and several of these key collaborators, and about whom I mentioned this work in this talk. So thanks a lot to all of them. And now in Australia, we are having really good collaboration with UWA, Coffee, Griffith, and DAFS, University of Sydney, ANU, University of Queensland, Agriculture Victoria, Western Sydney University, University of Tasmania. So a lot of good collaboration and a lot of funding. And we were really very privileged. We had really generous funding from large number of partners when I was working at ICRISAT. So thanks a lot. And thank you very much. And finally, that in the case if you are working on wheat, you are most welcome to join us 3rd International Wheat Congress in 2024. So you can come to Perth for PAG Australia this year, and then also that IWC for the 2024. Thank you very much. Happy to take a question.